Okay, guys. So my recording software was just doing weird things, so let's hope it works. Okay, so we're moving on to cell cycle. Who's excited about that? Who loves the cell cycle? I love cell cycle. You know why? Well, we wouldn't be here without it, but also because there's all these post-translational modifications that regulate cell cycle, and that's fun for me. Okay. Yeah. So let's move on. There's an exam this week. In case anyone hasn't realized that, for those of you who need to schedule an exam with the testing center, make sure we do that. For those of you who don't want to take the exam, you don't have to. You can just drop it. But then you have to take the other ones. Okay, so there was an error on the quiz this weekend. A horrible error, and this is what happens when you're writing a grant with one hand and writing a quiz with the other. You really make foolish mistakes. So thank you for those who emailed me about it. The upsetting thing is that only about 2 people emailed me about it, which means a really glaring error was completely, well, a lot of people got the question right, which concerns me. Yeah. It means either people are cheating, which we've had that problem in past classes where someone's like the sacrificial lamb and like fails it and then passes all the grades or all the answers around, and usually I can figure that out when I make these errors, which happens at least once or twice a semester because I'm not perfect. People get it right, but this one was an easy one to mistakenly get right. But I've had some in the past that have been really awful. And people have gotten them right. So, I'm just saying, I can see everything, and I see all of it. Yes? Oh, we're getting there. I know, we're very excited. All right, 2 points. Pretty sure it was the easy question. 2 points all around. Okay, so you benefit from my mistakes, because maybe you would have gotten it wrong anyway. I know, I'm sorry. Okay. So here was the wrong question, and you guys can let me know what is wrong about it. You're conducting a complementation assay in yeast that are Lu negative, meaning the yeast strain is Lu negative. You're studying telomere function by adding telomere sequences to linearized plasmids containing the Lu gene and growing them in leucine deficient media. That is your selection. Your experiment isn't working. Very, very little yeast, or no yeast, are growing in your leucine deficient media and normal media. Which of the following are plausible explanations for your results? Who can tell me what's wrong? No one who emailed me. Who can tell me what is wrong with this question? Yes?
I thought it was weird that the leucine deficient and the normal media had the same results.
Right, that was very weird. Yep.
So then I thought that it couldn't have anything to do with the mutated Lu gene.
That was absolutely right. Okay. What else? Did anyone else see something weird in this? Did anyone say, is anyone sitting here going, I think it's a fine question, what's wrong?
Yeah. I did because I kind of overlooked the distribution and media results, but in the lectures you put a lot of stress and emphasis on the ARS and the centromere. So.
Right, so that's where, okay. Yeah, so in my lectures I did stress the ARS and centromere, and I was trying to design this question to make sure you guys understood that some origin replication, which is what the ARS and centromere is required for chromosome segregation. But I was trying to make it a little extra tricky and basically messed up in the process. So the big thing is that in the normal media, everything should grow no matter what because it has nothing to do with the plasmid. Right? And because even though it's Lu negative yeast, it's normal media, it's not leucine deficient, so everything should grow. If you spit on it, something's going to grow. So thank you for pointing out. So let's fix this and see what we got. See if this works better, okay? You're conducting a complementation assay in yeast that are negative. You're studying telomere function by adding telomere sequences to linearized plasmids containing the LU gene and growing them in leucine-deficient media. Your experiment isn't working. No yeast are growing in your leucine-deficient media. Which of the following are plausible explanations for your results? You have no Rs. I'm going to say Rs. Would that result in no growth? Yes. Why?
Because
Go ahead. You need origin replication. If you don't have origin replication, then you're going to be diluted out fairly quickly. You have a mutated LU gene in your plasmid. Is that a possibility? Yes, if that results in not making leucine, right? Because then you're really leucine deficient. You have no centromere sequence in your plasmid. Is that possible? Yes. You forgot to remove leucine from your media. Is that possible? No, because you need to remove leucine. I mean, if you didn't remove it, then everything would grow. You would have a different result, right? You forgot to add leucine to your media. Okay. Okay, so some people feel better about this. Does everyone feel good? These kind of questions you should definitely be understanding at this point. Okay? So sorry if that freaked people out or if you spent too much time. Always email me if something doesn't seem right because there's a possibility it's not right. Okay, let's move on. Cell cycle. Woo! So I put this figure up. Is my introductory figure to cell cycle because normally I put the stereotypical figure showing DNA and actin or tubulin to show the mitotic spindles. But I came across this one and I thought it was very cool because SUMO is a ubiquitin-like molecule that's added to proteins just like ubiquitin. So it's a post-translational modification that affects a lot of things, but mostly localization and protein-protein interactions. And actually, there's a fair amount of work looking at SUMO and cell cycle. So I'm just going to walk you through the figure. You don't need to worry too much about this data. I just want you to get used to looking at immunofluorescence and get you used to just looking at data in general. So here they were looking at SUMO1 and SUMO2-3. They say 2-3 because they're both similar. And they're standing for two things. In the top, it's just a SUMO antibody. And in the bottom, they're merging it with DAPI, which is what? DNA. Even if you don't know, you can guess because look at what's happening over time here. And then SUMO2-3 is the green and blue is the DAPI. And so you can see, first of all, forget SUMO. Just look at DNA. Look at what happens. Interphase, not condensed at all, right? You don't have condensed DNA during interphase. Then prophase, and then you start to get more condensing. Well, I have to say, they did not do a very good job showing individual chromosomes in this picture. And then you get metaphase, anaphase, telophase, late telophase, as you start to go towards cytokinesis. What's really interesting about what they did is you can see SUMO1 is binding to SUMO1 and SUMO2-3 are binding to different places throughout the cell cycle, specifically in mitosis. You have a lot of SUMO1 binding to, what's this here? What do you think those are? Or what do you think this is right here? I don't know. You guys are not cell biologists. It's okay. Mitotic spindle, kinetochore. So it's involved in interactions between your chromosomes and your mitotic spindle. And SUMO2-3 is really bound quite heavily to also mitotic spindle. But look at how it's all over the DNA here, all over the chromosomes. So all I'm trying to show you is cell cycle, DNA changes. And that there are a lot of post-translational modifications involved in regulating it. And here I'm just giving you the example with sumo. What we're going to talk about through cell cycle is mostly the role of phosphorylation and the role of, because of kinases are going to be activated at different phases of the cell cycle, and the role of ubiquitin because we're going to be regulating the levels of cyclins. We aren't, the cells do. Yeah, so if you were a scientist, which you will all be at some point, right? Everyone here is going to be a scientist of some sort, right? Why not? And you were studying sumo and cell cycle, and you wanted to show that it was important for some phase of the cell cycle, what would you do? You already see that it's binding to all this stuff associated with changes that happen throughout the cell cycle. What would you do to your sumo proteins or your something else? Yes?
Could you remove them?
Could you remove them? Yeah, exactly. And that's exactly what they did in this paper, if anyone wants to look at it. They actually inhibited a different form of stimulation and showed that the cells got stuck at certain places in the cell cycle. Any questions about this? Okay. Okay, let's move on. So this is just an introduction. We're going to go over just some basics and then some methods as we move into cell cycle. And I'm going to use the cell cycle as the backdrop for explaining DNA replication and mitosis. Okay? Okay, so cell cycle is basically controlling, copying the genome and organelles and distributing to the daughter cell. This is straight out of 201, right? You need it for the survival of the organism. And you need it to for growth and to replace cells that die every day, right? So you've got cell growth, chromosome replication, chromosome segregation, cell division. Major elements of the cell cycle are chromosome replication and distribution to daughter cells, right? What phase is chromosome replication in? Actually, I have to stop just calling out people. Where is Daniel? Yeah.
Chromosome replication is in. Are you talking about DNA?
Yeah. Well, chromosome and DNA replication, yeah, same, interchangeable. What phase of the cell? And I haven't even introduced the phases because I'm expecting you know it because you guys already went through 201 and 301. What phase of the cell cycle does DNA replication happen in? Interphase? You are right, interphase. What, yeah, but what, okay. And so, yeah, it's interphase, you are absolutely right. But what part towards, there's a part of interphase. Yes? S phase, right. Yeah, S phase occurs towards the end of interphase. So you are absolutely right. And so then distribution to daughter cells, what's another word for that? Charles Hawkins? Mitosis, good. Okay, so DNA is replicated towards the end of interphase. If all goes well, the cell progresses to M phase, where duplicated chromosomes are segregated to the two daughter cells as a result of mitosis. I like to put this in here because people always think interphase is some part of mitosis, and it is not. Interphase is the part of the cell cycle that is not mitosis. So G1, G2, S, that's interphase. Mitosis is mitosis. And I want you to understand this because sometime I might show you something and I might say blah 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 during mitosis because things are different during mitosis, or I might say blah 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 during interphase. So understand that because on an exam you don't want to answer for mitosis when I'm talking about interphase and vice versa. I'm just going to highlight that. Okay, so this is a classical cell cycle figure that you guys have seen a million times. especially if you have me last semester. And it's just showing that there's these gap phases between S and M. Ha ha. Giggle, go ahead. Giggle, giggle. Okay, next time I say M and F. I know we're all like 13-year-old boys, aren't we, when it comes down to it? Okay. So what is the purpose of these? What is the purpose of these two gap phases, right? So here's G1 and here's G2. Where is Sean? Yes. What's the purpose? I know this is great because when you start zoning out, you'll be like, whoa, someone said my name. So there are these gap phases between S phase and M phase. What's their purpose?
Well, checkpoints going between them, check to see if the cell dividing correctly or is replicating correctly.
Okay, so there are checkpoints involved with these gap phases to make sure the cell is functioning properly. What else? Let's try for Shawnee. Okay, so what needs to happen before S phase? We can raise hands. Yes.
So, I mean, the chromosomes grow.
Before S phase, cell needs to grow. Cell needs to grow. So all of this needs to happen before M phase in general. The cell needs to grow quite a bit. Remember, because it's going to split into two. So there's a lot of cell growth that's happening. What needs to happen between S phase and M phase? Give this page some attention. Hawa. So there's a checkpoint. What kind of checkpoint? What are we looking for? Right, so to make sure S phase has completed, right? Because if there's DNA damage, that's going to stall replication forks and you're not going to complete S phase properly. There could be some other damage. Okay, good. What about G? So there's this G0, which is not on here, and it occurs before S phase under certain conditions and certain cell types.
Isn't that when the cell's not going to divide? It's just going to stay as a single cell.
So right, the cell's not going to divide because it needs an extra amount of time either to grow or wait for the environment to improve, or certain cell types live in G0. What cell type do you think about live in G0? This would be a cell that doesn't divide. Yes. Brain cells. Yeah.
Can we induce G0? Can you induce a cell to go through the cycle?
Can you induce cell cycle? In the lab or in anywhere. In the lab, absolutely. Anywhere, I don't know. You really don't. So cell cycle is tightly regulated because when you go into S phase and you shouldn't, then that's cancer. So I don't know. I know there's a lot of ways to halt the cell cycle, but I'm not really sure. I can't think off the top of my head of ways. So are you trying to think of ways to grow cells that aren't growing or something like that in terms of regeneration? Yeah. I have to think about that one a little more. Okay. So I keep saying cell cycle is tightly regulated. There's a bunch of proteins, and they control a cell cycle, and they act as these biochemical switches that allow the cell to progress to the next phase only if the previous phase was properly completed. And so these you can think of as checkpoints, right? And so I already have someone mention checkpoints. So there are three main checkpoints. The first one is start, and this says, are we ready? Is the environment favorable? Are we ready to start replicating our DNA? Then there's a checkpoint before you enter mitosis because you don't want to divide your cells if there's a lot of movement today in this class. I keep seeing people getting up and moving. And people are freaking out about the test. They're like, I can't sit here. I need to go study. Should I just drill you guys on an exam question? I know, that would be cool if I did that. I feel so nauseous. You know that my daughter came home on Friday and puked SpaghettiOs all over the stairs. And that continued until 3 in the morning. She was better by Saturday night, and all is well. And I have to say, I've become a professional in infection control because the other children have not puked. Feel a little nauseous myself. Would it be weird if I puked right while I was lecturing? All over the place. It would be disgusting. Okay, sorry, I digress. Like sometimes you can't help but stop and voice what's going through your mind. Usually I don't voice my grocery list while I'm teaching you, but when it's this overwhelming need to throw up. Okay, anyways, I'm not going to throw up, I promise. Maybe I'm just nervous for you guys about the exam. Anyways, okay, so then there's another checkpoint, metaphase to anaphase transition. And this is one just to make sure that all the chromosomes are appropriately attached to the spindle. If they're not, that's a big problem, right? If you don't have all your chromosomes attached to your mitotic spindle, then you're not gonna segregate them properly. You're gonna end up with uneven chromosomes in each cell. So those are the 3 main checkpoints. Okay, so yes.
If an error is detected, are there corrective measures?
It depends what kind of error. Yes. If an error is detected, are there ways to correct it? Absolutely. There's a whole DNA repair system, which we'll learn about towards the end of the semester. I probably should put that right after this, shouldn't I? And then in terms of mitotic spindle attaching to chromosomes, there's this whole system for detecting if the correct tension is there, and if the correct tension is not there. So like if your mitotic spindle on one side attaches to both sister chromatids, the tension won't be right compared to like each side attaching to one sister chromatid. And there are mechanisms to prevent that attachment, and actually it results in detachment and it allows it to go again and again and again until it's correct. And I'm guessing if at some point things just aren't right, then I don't know, then it's not going to move forward, right? Okay. So cell cycle is regulated by many different proteins. Why is cell cycle regulation so important? Why am I saying this again and again? Where's Miranda? Yes. Why is cell cycle regulation so important?
Because you don't want anything to go out of control, cancer, disease prevention, and regulation.
Right. You don't want cells to grow out of control because you might end up with cancer. Right. Absolutely. So, how do cell biologists study cell cycle regulation? So I'm going to give you a few different methods, common methods. This was very cool because I was writing my grant and I wrote in it, blah, 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 will study effects on cell cycle and apoptosis. And in the last round, they came back and they were like, how? I forgot to put in how. So this time I was like, oh, cool, I can do PI staining, all the things I'm about to teach you, I will be doing if I get this grant funded. So it's very exciting. Okay, so the machinery involved in cell cycle regulation is highly conserved throughout eukaryotes. So much of the research that's led to what we currently know about cell cycle came from different model organisms, such as yeast, animal embryos, and cultured mammalian cells. And you guys already had a little bit of an experience with studying cell cycle and yeast in a way, right? Because you guys were looking at centromere DNA sequences. Okay, so yeast are a commonly used model system due to ease of genetic manipulation, mostly. There are two kinds. There's fission yeast and budding yeast, and it just has to do with the fact that they divide differently. So you can study cell cycle control in yeast through analysis of genetic mutants that are defective in cell cycle progression, right? So you make all these mutants that have some sort of defect, and then you can do these kind of complementation assays to study. So I'm going to introduce the concept of conditional mutants to you guys. And conditional mutants are important because when you're studying cell cycle, if you have a defect in a cell cycle protein, you can't study it if it's defective because you can't propagate those yeast. Does that make sense? So they make these conditional mutants that are only expressed under certain, or the defect is only seen under certain conditions. And so the example I'm going to give you guys is temperature sensitive mutants, which at the permissive temperature, the protein works fine, and then when you raise the temperature to a restrictive temperature, you can actually visualize the phenotype of the defective protein. And how this works is usually there's a slight mutation that might involve folding of a protein or something like that. So when you raise the temperature, the protein becomes misfolded, and it doesn't work properly. And you can actually engineer these mutants. So normally, under permissive temperature, your cells go G1, S, G2, M. And if you've got a defect, you'll raise the temperature, and what will happen is they'll all go until they hit the point of the defect. And in this example, the defect is somewhere between G1 and S. You can have temperature-sensitive mutants, and I think there's some. What's the other? There's another. You can have mutants where you induce them under certain by adding a chemical or a drug or something like that. Yes. pH-sensitive mutants? I don't know. Maybe. I haven't heard of them, but I follow your logic. Because pH is involved with structure of protein. Yep. Yes?
How do you ensure that when you're raising the temperature, you're only disrupting one protein and not all the proteins?
How do you know when you raise the temperature, you're disrupting that one protein and not all of the proteins in the cell? Right, that is a concern. And so usually the high temperature is not that high. You'll go from 25 to 37. And these mutants, so there are these libraries of mutants that have been well characterized to only show a certain defect. They show a very specific defect under certain conditions. So if your cells die, you wouldn't have that mutant. So you need something that you're able to see a very specific defect, such as the ability of the cells to divide, or well, I'm gonna actually show you an example in just a minute. Any other questions? Okay, so here they're using budding yeast to study defects in CDC15. And so they're budding yeast, so they're not going to divide in half. You're gonna see a bud form, right? So here you can see in wild type, they're at all these different phases, because remember, cells they don't grow in a synchronized way. They're at different phases of the cell cycle at any given moment, unless you synchronize them. So here, these are pretty much about to split. Here's a bud just forming on this one. Here the bud's a bit bigger. Here this one's not doing anything. So this is normal. These cells are normal, right? And when you grow these same exact cells at the restrictive temperature, and they're the CDC15 mutant, then you see they all end up synchronized at the same place. So they all started like this, and they moved forward in the cell cycle until they hit the point where there was a defect. So what do you think the defect is here? Oh wait, let me call someone. Jessica, Mickey.
They can't, cells can't separate.
Okay, so something is wrong with cytokinesis. Yep, the cells can't separate all the way. So based on that, and based on what you know about cytokinesis, which isn't much, but you can probably think about it a little bit, what do you think CDC 15 might be doing? Vito. Something with the mitotic spindles. So you think that the metaphase anaphase transition checkpoint might be triggered? Maybe. Though I would suspect we've gotten past that because we're even past that almost to cytokinesis, right? But that's good because you're towards the end of mitosis. Yes.
Negative burden of actin is going on the cell.
Something about actin. Something about actin. And actually, you're right. That is, I looked it up because I wanted to be sure I knew. And yeah, it is CDC15 is a kinase that's involved in reorganization of actin to create that contractile ring that is involved with completing cytokinesis. Yeah. Any questions about this? Pretty cool, huh? Don't you guys all wish you worked on yeast? I highly recommend. Yes. Huh? Well, this. So you're wondering if the cells on the right will continue to function after? No, they're stuck right there. They're stuck. You could turn the temperature back down, let them recover, but at the restrictive temperature, that's where they're. So you only raise that temperature when you're looking to study the defect, and then they die, and that's the end of that. Yep.
So if you raise the temperature and the slide you showed previously doesn't get past the G1S checkpoint, how do you also know there's not a mutation in something occurring in cytokinesis as well? Would there be a mutation earlier in the pathway than later as well?
So your question is okay, so you raise the temperature, you get past the G1S checkpoint, you go through cell cycle, and then we stop here. How do we know the mutation's not earlier as well?
No, well, if the mutation would be earlier, like they wouldn't end up replicating the DNA, they would be stuck in G0 at that point. How do you know that there's not also a mutation that was caused that could affect it later?
Okay, so you're saying if you see a mutation early in the cell cycle, how do you know there's not another one that's later?
Yeah, like this one.
Right. Okay. Yeah, you're right. I think you're stuck characterizing the one the phenotype that you can see, and there might be multiple mutations involved, and that would make it more complicated. Yeah. Any other questions? Okay. Okay, so, so I pulled this figure to show you that it was involved in actin organization. And this is from a paper in 1995 where they were looking at CDC15. And here's DAPI, so that's just the nuclei. You see they're about to divide. And this is not budding yeast. Notice there's no way. This is fission yeast. So you can see the two nuclei. Oh, I don't know what's wrong there. It's got to be something else. And they're looking at different mutants, CDC7, CDC11, CDC14, and CDC15, and this is the one we care about. And they're staining actin, and they're looking at, so if CDC15 is working properly, you should see the actin contractile ring forming in the center, and it's forming normally, normal, I don't know what that is. And look, it's not forming normally here. So that was the defining figure in that paper. that suggested that CDC 15 was involved in contractile ring formation. Yes?
You said actin just essentially completes cytokinesis?
There's an actin contractile ring a contractile form of actin that forms in the mid-zone of the cell that's about to divide. And it pinches, yeah. If you take cell biology, you'll learn about that in gory detail. Not that gory, right? We actually spent a little bit of time on that part, but we did spend a lot of time on actin and regulation of that. Okay. Any questions about this? Pretty cool, huh?
The other CBCs are just other mutants.
Other mutants, and you can see something's going on with them, right? Not with actin. So but if you look at DAPI, something funny is here because there's only one nucleus. So I would guess that CDC14 has. I don't know what they all are doing. CDC stands for cell division cycle mutants, and they just named them. Like they just numbered them as they came across this different defects.
But those mutants actually make it towards mitosis.
Those mutants make it towards mitosis.
The mutation doesn't stop them like SMAs.
Yeah. Looks like it. Yeah.
Don't you have a lot of multi-nucleate fungi though? So can they still be functional cells?
We have multinucleate. So these are cells that are about to go into cytokinesis.
Right, but if they stop and they're not able to actually complete cytokinesis, can they still exist as multi-nucleate cells?
Can the yeast still exist as multinucleated cells if they do not complete cytokinesis? I don't think so.
Some type of fungi are multi-nucleate, so I didn't know.
Yeah, I don't know. That would be this would be a really bad model system if they had ways to get past it, right? Okay. Any other questions? Great comments today. Okay. So that was yeast. Immortalized cell lines can grow indefinitely. And they represent a tool for studying cell cycle as well as many other areas of cell biology. So what are immortalized cells? Does anyone know what I mean? Kelly. Okay. Michael? Where are you? They keep growing, right. So normal cells, if I take them out of your body, will only divide 50 or 60 times, and then they enter senescence, basically, and then eventually die. But immortal cell lines, so cell lines that are using cell culture for the purpose of research, they replicate indefinitely. How come? Have you ever thought about why certain cell lines replicate forever and certain cell lines don't? Yes?
It has to do with
Telomerase activity. So some cells do, one way of making an immortal cell line is to add in high-level telomerase expression. Yep, that's one way. Yep.
Can't they infect certain cell lines with SB40 virus?
Right, so some cell lines are infected with viruses or have parts of viral genomes integrated to make them immortal, right? So SV40 is one. Yep, adenovirus is another. Yes?
Some of the cell lines cancers like HeLa cells, so they don't have to go through regular checkpoints.
Right, so some cell lines are cancer cells, so by definition, they can replicate forever. HeLa cells are a great example, and that actually is cancerous and virus infected, right? Yeah.
Could you treat the cells with a growth factor?
Could you treat the cells with a growth factor? Normal cells you do not want to treat just with a growth factor because if you have unregulated signaling, sometimes you end up triggering like P53 or different genes that are supposed to stop cell cycle. So, but a lot of cell lines, specifically human umbilical vein endothelial cells. I only know because I've worked with them, that are very difficult to work with, require a fair amount of growth factors, but they've been transformed also. Okay. Good. Okay, so different tools for studying cell cycle. Well, there's the most obvious, which is just look at them. Visualization. You can directly look at them because cells in mitosis are grounded. Cytokinesis is pretty obvious to notice once you start to look at cells. If you see two that are coming apart, probably cytokinesis. You can also use DNA stains or antibodies to visualize cells at different phases of the cell cycle. This one here is microtubules is in green, and DNA is in DAPI, which is blue, so that's the DNA stain, and the microtubule. Microtubules become the mitotic spindle. So you can differentiate between different phases of the cell cycle just by looking for the organization of your chromosomes and your mitotic spindle. So these cells, what phase of the cell cycle do we think these ones, right? So we've got 1, 2, 3, 4, 5, 6, 7 cells in the field. These cells are clearly, these two are clearly different than the others. What phase of the cell cycle do you think the others are in? Katie. Katie. Yeah.
Which one's?
Huh?
Which one is
So this one, this one, this one, this one, and this one are in a different phase than these two. What phase do you think they're in? Yeah. Interphase, right. Interphase, because those of you who've taken cell will have learned that the microtubule network changes a lot from interphase to mitosis, but also just looking at the DNA, you don't see any condensed DNA, right? It's just, this is interphase nucleus. And here you actually see your chromosomes that are condensed, and you're in two different phases of the cell cycle here. What phase do we think this is? Jackson. Prophase, right? Because you've got condensed chromosomes, but they're not lined up yet. Compared to here, which would be in what? Kristen. Yeah.
Yeah, it's the one that lines up in the end. I don't remember the name.
Metaphase. It's okay. Metaphase, right. They line up right in the center. Yep. So there's a lot that you can learn just from staining your DNA, yes?
So by the intensity of the DAPI stain, can you tell which ones have undergone S phase and which haven't?
We're getting there. By the intensity of the DAPI stain, can you tell which cells have undergone S phase and which can't? No, not with, probably, actually, let me take that back. Probably, but that's not the traditional way people do that. But you just gave me a perfect segue to where we're going. Because there are two ways to look at cells, and determine whether they've gone through S phase recently, and also a way to look at DNA content and determine where they are in the cell cycle. So one, see, that was perfect. I did not plant that comment. Though I might start planting comments. Thinking about that. Cells in S phase, they can be identified by detecting incorporated base analogs with specific antibodies. What does that mean? A base analog. Well, I put up a picture here. What I'm talking about is BRDU, which is a thymidine analog. So anywhere you'll have a T, you put the BRDU will be incorporated during DNA replication, and then you can use an antibody that recognizes it to detect it. So here's your normal thymidine, and here's your BRDU, and the only difference is you've got this bromide group added right there. So this picture is showing BRDU labeled epithelial cells in a zebrafish gut. It's right out of the textbook. The anti-BRDU is green, which looks yellow here because it's overlapped with red. And all cells are stained red with like an actin stain or something like And so all of these cells here that are yellow have recently undergone S phase because when they're replicating their DNA, they've integrated the thymidine analog. So anytime that you need to put a T in, they put the BRDU in. Then you take your cells, you put them on a slide or however you want to count them, and you add primary antibody, secondary antibody that gives it some color, and you can detect them that way. This way in the microscope. You can also detect them using flow cytometry, which I'll introduce in a minute. Any questions about this method? Yes?
I'm kind of confused about, do you want the BRDU or taking it out or?
You're using it, so these are cells that you're just using in your lab to study something. So let's say you're studying some protein and you think it's involved in completion of S phase or some part of cell cycle. And your question that you ask yourself is, well, if I mutate this protein, will S phase happen or not? And so this is a good way to answer that question. You take your cells that are growing in cell culture and you add BRDU to them and let them go through DNA replication. And then you harvest them and you treat them with antibodies so that you can detect them. And you can say, oh, well, all these cells went through DNA replication. It must not be a problem there. Make sense? Any other questions? Yes?
I may have to send this, but do you know exactly how they incorporate the analog?
You just literally, like, put, however many microliters in your cell culture media.
Really?
Yeah.
Is it
They don't-
isotopic bromine?
This is just, no, it's just normal, and it's not radioactive. And you detect it because there's an antibody that will recognize it. So when you have your cell, you can take your cells out of culture, you can plate them on a slide, and then you could permeabilize them by treating with some detergent so that pokes holes in all the membranes. And then you add your antibody, it can get into the cell, into the nucleus, and bind to the DNA that contains these analogs. Okay? Immunofluorescence is fun, isn't it? We just bought a $35,000 awesome microscope in this apartment to do all this kind of fun stuff. It's very exciting. I'm excited. Okay. I say $35,000 because who knew that it cost that much? That's actually a cheap microscope. Okay. So flow cytometry, I just mentioned flow cytometry, and in the last few minutes I'm going to introduce it. This is just a picture of what's happening inside a flow cytometer. But flow cytometry, you can think of as a way to count cells. It's a way to count cells based on whatever characteristics you want to count them on. You can label your cells with BRDU and count all your green cells, or all your red cells, or all your whatever color you choose. You can separate a population of cells based on shape and size. If you take all the cells out of your blood, you'll have many different cell types, which will all have different shapes and sizes, and you can separate them based on that. So the way it works is you pour your cells into the top here, and they drip down through they drip down in a stream, and then you have lasers that are directed at the cells. And then you've got these mirrors that capture the way the lasers bounce off the cells. And you can think of it as light is going to bounce off objects in ways that are dependent on the shape, right? The shape of the object. So you can capture things based on their shape that way by looking at how the light scatters off of the cells as they pass through. You can also detect different fluorophores. You want to detect green things or red things or whatever, and then you end up plotting things on. So it's detected by a computer, and then it gets plotted in a graph of some sort. And so this is what it ends up giving you. So, oh man, are we running out of time? A little bit. Okay, let's start with flow cytometry on Wednesday. Good luck to everyone. Amazing.